Welcome back to the deep dive. Today, we are really getting into the weeds on a major medical challenge, septic shock. This isn't just about a bad infection. It's about what happens when the body's fight against that infection goes haywire, becoming, well, self-destructive.
Yeah, it's the ultimate, you could say, circulatory failure. It leads pretty much inevitably to cellular hypoxia cells not getting enough oxygen. And, shock has different forms, right? Cardiogenic, where the heart's the issue, hypovolemic, not enough fluid. But today, yeah, septic shock is our focus. It's definitely the most complex because it's not just a mechanical problem. It's a systemic molecular meltdown, all driven by that immune response going off the rails.
Exactly. And that's our mission for this deep dive. We want to get past just the symptoms and really understand the pathogenesis. How does the body's own defense system actually create this life-threatening crisis down at the level of cells and blood vessels?
Okay, so to really get started, we need the latest definitions. For a long time, sepsis was, frankly, a bit confusingly defined. But the 2016 international consensus really brought some much-needed clarity.
Okay, so what does that latest framework actually tell us? And why is the wording so critical?
Well, it fundamentally shifted the focus. Sepsis is now defined very clearly as life-threatening organ dysfunction caused by a dysregulated host response to infection. And the key phrase there, the absolute core of why we're doing this, is dysregulated host response. It's not just about the microbe invading your system. It's the body's reaction spinning wildly out of control, an overreaction that's arguably worse than the initial threat itself.
Right, so if sepsis is that organ dysfunction kicking in, where does septic shock fit into that picture?
Septic shock is, well, it's the most dangerous subset of sepsis. It's specifically defined by having these really profound circulatory, cellular, and metabolic abnormalities. When a patient meets those clinical criteria, we know their risk of dying has just skyrocketed, even with everything we try to do.
And the scale of this problem, wow. It really hits home when you see the numbers. Our sources mention septic shock is behind something like 2% of all U.S. hospital admissions. That's huge. Over 750,000 cases a year.
And that's probably lowballing it globally. What's really sobering is that despite all our ICU advancements, better monitors, stronger antibiotics, life support, the mortality rate, it's still stuck stubbornly around 50%.
Yeah.
50. It's a battle we still lose about half the time.
And here's a twist, right? The number of cases seems to be increasing. How does that work when medicine is generally getting better? Why more cases, and who's most vulnerable?
Yeah, it seems counterintuitive, but there are a few key drivers. First, ironically, better life support keeps very sick people alive longer, but they often have underlying issues that make them susceptible later on. Second, we just have way more immunocompromised people now. Think about the aging population, widespread chemotherapy use, patients with HIV, or complex autoimmune diseases needing strong drugs. Their immune systems just can't mount a clean, regulated defense.
So it's either too weak or it just goes completely haywire.
Exactly. It's an uncontrolled response. And the third big factor, you hear about it all the time, multi-drug-resistant organisms, MRSA, VRE, all those. If the initial bug causing, say, pneumonia or a UTI is resistant to our go-to antibiotics, it gets a head start. That gives the host response more time to, spin out of control before we can intervene effectively.
Okay, so what are the usual suspects? Which microbes typically trigger this whole mess?
Well, almost any pathogen can, technically, but most often it's gram-positive bacteria, things like staph or strep. After that, you see gram-negative bacteria pretty commonly, and then less frequently, fungal infections can do it too. But that diversity is actually really important. It tells us the real problem isn't some specific toxin from one bug. It's the generalized activation of the body's own defense systems.
That sets the stage perfectly. Let's get into the core of it. The dysregulated host response, starting with section 1. The inflammatory ignition, how we get from microbe to this meat-eater storm.
Right. So it all starts with detection. Your immune system is constantly scanning, looking for trouble. It recognizes invaders by these conserved molecular signatures called pathogen-associated molecular patterns, or PAMPs. So, for instance, gram-negative bacteria have lipopolysaccharide. LPS, you might know it as endotoxin. That's a classic PAMP. Gram-positives have things like peptidoglycan. These are like the microbial barcodes.
And how does the body actually read these barcodes? What are the sensors?
It uses these specialized receptors called pattern recognition receptors. The sources highlight several key families. Probably the most famous are the toll-like receptors, the TLRs. They recognize a huge range of microbial stuff. TLR4, for example, is the big one for sensing that gram-negative LPS. But there are others too, like G protein-coupled receptors that detect bacterial peptides, and C-type lectin receptors like dectins, which are really important for spotting fungi.
Analogy time. So these receptors, they're like the tripwires and motion detectors on the immune system's fence. When they sense a PAMP, they don't just send a quiet note, they hit the big red alarm button, screaming, "Intruder!"
That's a great way to put it. And it's not just your classic immune cells like macrophages and neutrophils that have these sensors. Even the endothelial cells, the ones lining your blood vessels, they've got them too. And crucially, it's not just PAMPs from microbes. The sources also point out DAMPs, damage-associated molecular patterns. These are signals released from our own cells when they get damaged or die. Things like mitochondrial DNA getting out. These DAMPs also trigger the same alarms, adding fuel to the fire even if the initial bug is being dealt with.
Okay, alarms triggered. What's the internal command system that launches the full-scale inflammatory war?
That alarm signal triggers this complex chain reaction inside the cell, ultimately activating the master switch for inflammation, a transcription factor called nuclear factor Kappa B, or NF key. You really have to think of NF key as the key messenger. It rushes into the cell's nucleus, binds to the DNA, and basically tells the cell, okay, start mass producing all the inflammatory weapons now.
So it's like the foreman flipping the main breaker in the cytokine factory.
Exactly. Once NFP is activated, it dramatically ramps up the expression of hundreds of genes that code for inflammatory mediators. This is where that cytokine storm really gets its start. The sheer volume and speed just overwhelm the body's normal checks and balances.
Okay, walk us through the main players in this mediator storm. What are the primary destructive agents being unleashed?
Well, the first wave is really led by the big guns, the major pro-inflammatory cytokines, tumor necrosis factor, TNF, and interleukin-1, IL-1. Think of them as the field commanders of the storm. They're responsible for causing fever, activating neutrophils, kicking off that endothelial damage we'll talk about. They're really central to making this a systemic problem, not just a local one.
What else gets deployed in that initial onslaught?
We also see a surge in IL-12, IL-18, and interferon gamma, IFN. These tend to amplify the immune response further, calling in even more reinforcements. And one mediator our sources specifically highlight is high-mobility group box-1 protein, HMGB1. Interestingly, this is actually a DAMP. It's released from our own dying cells, but it acts like a late-stage pro-inflammatory cytokine.
Why is that timing important? The fact that HMGB1 is late stage.
It's really significant clinically because it means the inflammation can keep going, can persist, even if we manage to kill the initial bacteria with antibiotics. The body is still reacting to its own internal damage signals. This actually makes HMGB1 a really interesting potential target for new therapies trying to shut down that self-perpetuating damage cycle long after the initial trigger is gone.
And this whole overwhelming storm of signals, we can actually measure it, right, in patients.
Oh, absolutely. This is why clinicians track markers like C-reactive protein, CRP, and procalcitonin. They shoot up during this acute inflammation. But beyond just the cytokines, the storm also includes other damaging molecules. Reactive oxygen species, ROS, those cause oxidative stress, and potent lipid mediators like prostaglandins and platelet activating factor, or PAF. These guys really add fuel to the fire, especially accelerating the damage to the blood vessels.
Now, we can't discuss a big immune fight without mentioning the complement system, that ancient cascade of proteins. How does it get tangled up in septic shock?
Right, the complement system. It's a cascade of proteins normally floating dormant in your blood. Microbial components, PAMPs, again, directly activate it, often through what we call the alternative or lectin pathways. This activation rapidly generates some really potent fragments, particularly the anaphylotoxins C3A and C5A, and opsonins like C3b, which tags microbes for destruction.
And these fragments, C3A and C5A, they just make everything worse in this context.
Oh, absolutely. They are powerful pro-inflammatory drivers in their own right. C3A and C5A cause mast cells to degranulate, releasing histamine and other stuff that increases vascular permeability, making vessels leakier. Plus, they're incredibly potent chemoattractants. They basically summon huge numbers of neutrophils to the area, which, while trying to help, end up releasing their own toxic enzymes and ROS, causing significant collateral tissue damage.
Okay, before we pivot to the blood vessels themselves, I want to circle back quickly to superantigens, because that's a really stark example of a different mechanism causing the same catastrophic outcome, right?
It's a fascinating and, frankly, terrifying parallel. Some bacteria secrete these proteins. The classic example is the one causing toxic shock syndrome called superantigens. Now, normally a T cell needs a very specific antigen presented to it to get activated. Superantigens bypass all that. They act like a master key, directly linking the antigen-presenting cell to huge numbers of T cells, activating them non-specifically. Maybe 20% of your T cells all at once?
Wow. That sounds like hitting the launch button for all the cytokine missiles simultaneously.
Pretty much. It causes an immediate, overwhelming, and totally uncontrolled release of cytokines from all those activated T cells. Functionally, it mimics the entire septic shock cascade: vasodilation, leaky vessels, hypotension, shock S, but it's triggered by this massive direct T cell hyperactivation, not the typical PAMP-TLR route. It really underscores the central point. The host response, however it gets triggered, is the ultimate destructive force here.
Okay, so that chaotic storm of mediators perfectly sets up Section 2. Endothelial dysfunction and hemodynamic chaos.
Yeah.
The actual lining of the blood vessels starts to break down.
Yeah, this is where the molecular chaos translates into circulatory collapse. The endothelium, that single layer of cells lining every blood vessel, becomes a primary target. It gets bombarded by those cytokines we mentioned, TNF, IL-1, plus complement factors like C5a, and even directly by microbial products sometimes. This widespread endothelial activation and injury is what drives the profound circulatory changes we see in shock.
Let's break that down. First, vascular leakage. What exactly happens when those connections between endothelial cells fail?
The endothelium basically loses its barrier function. The tight junctions, the molecular glue holding the cells together, they loosen up significantly. This makes the capillaries, the smallest vessels, dramatically more permeable or leaky. And what leaks out isn't just water, it's protein-rich plasma fluid. Albumin, other crucial proteins, they pour out of the circulation into the surrounding tissues, the interstitial space. This accumulation is what we call protein-rich edema.
And this fluid leaking out everywhere, the systemic edema, that causes major problems for getting oxygen to the tissues, right? Even before blood pressure drops.
Absolutely. It creates a physical barrier. Think about it.
Yeah.
That extra fluid accumulating between the capillary and the tissue cell increases the distance oxygen and nutrients have to diffuse across. It seriously impedes delivery. And it also blocks the removal of waste products like lactic acid. So even if the heart's pumping okay, and even if we're pouring IV fluids into the patient, a lot of that fluid just ends up leaking out into this third space, making the edema worse and actually hindering tissue perfusion.
And while that's happening, the whole system starts losing pressure control.
Right. This is the massive vasodilation and systemic hypotension piece. Those activated, inflamed endothelial cells start producing huge amounts of nitric oxide, NO. NO is an incredibly potent, very fast-acting vasodilator. It signals the smooth muscle cells wrapped around the blood vessels to just relax completely. Combine that NO effect with other vasoactive mediators floating around like C3a, C5a, PAF we mentioned, and you get this catastrophic system-wide drop in vascular resistance. The pipes just go wide open.
And this profound vasodilation is kind of the hallmark of septic shock, isn't it? It explains that weird warm shock phenomenon you sometimes see early on.
Exactly. In most other shock types, like cardiogenic or hypovolemic, the body desperately tries to compensate by clamping down peripheral vessels. You get cool, pale, clammy skin.
Yeah.
But in early septic shock, that overwhelming NO-driven vasodilation can actually override those compensatory mechanisms. So the patient might initially present with warm, flushed skin, especially in their extremities. They technically have enough blood volume, maybe even increased cardiac output initially, but there's just no pressure, no tone in the system to push it effectively through the organs.
But the real reason things spiral downwards, the functional catastrophe, is at the smallest level, microvascular dysfunction. The capillaries, where the actual oxygen exchange happens, they just stop working properly.
This is the crux of the oxygen delivery problem, the mismatch. It's not just about the overall blood pressure. It's about where the blood is actually going at the micro level. In sepsis, a couple of key things go wrong down there. First, blood flow becomes really erratic, really heterogeneous. Some capillaries might shut down completely, have no flow or just intermittent flow. Others might have blood rushing through way too fast. It's patchy.
So oxygen isn't getting to some areas at all, and in others, the blood is moving too quickly for oxygen to effectively unload into the tissues.
Precisely. And the second, perhaps even more devastating issue is the complete loss of autoregulation. Normally, local tissues control their own blood flow. If a group of cells is working hard and needs more oxygen, the nearby arterioles dilate to increase flow to that specific spot. In septic shock, that fine-tuned local control is totally obliterated. It's overwhelmed by the systemic effects of NO, acidosis, and all the other inflammatory mediators.
Wow. So oxygen delivery becomes completely uncoupled from actual tissue needs. It might be going to tissues that are resting, while vital organs screaming for oxygen are being starved. A total mismatch between supply and demand right at the cellular front line.
Yes, and that profound failure of effective tissue perfusion, coupled with the systemic low pressure, really pushes the patient towards the final devastating phase, the fatal triad of coagulation, metabolism, and organ failure.
Okay, let's tackle coagulation first, because it's not just that the system gets leaky and floppy, it also starts clotting like crazy.
Oh, absolutely. The sources are very clear on this. The coagulation system goes completely haywire,
Oh,
so
yeah.
much so that up to half of septic patients develop disseminated intravascular coagulation, or DIC. And DIC is exactly what it sounds like, a life-threatening, widespread activation of clotting that leads to tiny thrombi, tiny clots, forming in microvessels all over the body.
Okay, walk us through how that happens. How did that inflammatory storm kick off this massive clotting cascade?
The main trigger appears to be tissue factor, or TF. Those pro-inflammatory cytokines, especially TNF and IL-1, cause monocytes and, importantly, those activated endothelial cells to dramatically increase their expression of tissue factor. And tissue factor is basically the initiator, the main on switch for the extrinsic pathway of coagulation. It signals massive injury detected, start clotting everywhere now.
So the body slams the accelerator on clotting. What happens to the brakes?
At the same time, the body essentially cuts the brake lines. Sepsis makes those same endothelial cells decrease their production of key anticoagulant factors. This includes things like thromboembolin, which normally works with thrombin to activate protein C, a major brake, and also protein C itself. We also see less tissue factor pathway inhibitor, TFPI. So with the natural brakes failing, the clotting just runs rampant.
And if that wasn't bad enough, the system for cleaning up clots also gets shut down.
Exactly. That's the disruption of fibrinolysis, the process that normally dissolves fibrin clots. Cytokines, particularly IL-1 and TNF again, stimulate the release of plasminogen activator inhibitor 1, or PAI-1. PAI-1's job is basically to block the enzyme that breaks down fibrin. So you're forming clots way too fast, the brakes aren't working, and the cleanup crew is inhibited. The clots just build up.
And there's a relatively newer player involved here too, right? These neutrophil extracellular traps or nets.
Yes, that's a really important recent understanding. Neutrophils are main bacterial killers. When they die fighting infection, they can undergo this process called netosis. They essentially spew out these web-like structures made of their own DNA, histones, and toxic granule proteins. These nets are great for trapping bacteria, but they also act as a physical scaffold and a powerful chemical catalyst that promotes coagulation, activating both the intrinsic and extrinsic clotting pathways. They really pour fuel on the DIC fire.
Okay, so you combine hyperactive clotting, failing anticoagulant systems, inhibited clot breakdown, and these nets providing scaffolds. You get clots forming everywhere in the small vessels.
Yes, widespread microvascular thrombosis. These fibrin-rich clots physically block blood flow in tiny vessels, worsening the tissue hypoxia we already talked about. But here's the terrible, terrible paradox of DIC. This runaway clotting is so aggressive, it starts consuming platelets and coagulation factors faster than the body can make them. This is called consumptive coagulopathy. You literally run out of the raw materials needed to form clots.
So the state of extreme clotting flips violently into a state of extreme bleeding risk.
Precisely. You might have microclots shutting down blood flow to your kidneys and lungs, but simultaneously, the patient is profoundly thrombocytopenic and lacking clotting factors, putting them at huge risk for, say, a catastrophic brain hemorrhage or uncontrollable bleeding from an IV site. It's the body attacking itself from both sides of the clotting system.
Unbelievable. Okay, moving to the second part of that fatal triad, the metabolic derangements. Primarily, this involves hyperglycemia and lactic acidosis. How does the inflammatory environment throw metabolism into chaos?
Well, the stress of severe sepsis almost immediately triggers hyperglycemia, high blood sugar, and insulin resistance. The drivers are familiar. Those same inflammatory cytokines, TNF and IL-1, working together with stress hormones like cortisol, catecholamines, adrenaline, and glucagon. These hormones crank up glucose production in the liver, gluconeogenesis, and breakdown of glycogen, flooding the bloodstream with sugar.
And the cytokines actively interfere with insulin's ability to manage that sugar.
Yes, they directly impair insulin signaling in peripheral tissues like muscle and fat. They can mess with the function of GLUT4, the main transporter that moves glucose out of the blood and into those cells. So you end up with the situation where the blood is full of glucose, but the cells that desperately need fuel can't take it up effectively because of insulin resistance.
And this high blood sugar isn't just a side effect. It actually harms the immune response itself, doesn't it?
It absolutely does. Sustained hyperglycemia is known to impair neutrophil function. Their ability to move, engulf bacteria, and kill them is reduced. So the high sugar actually weakens the body's fight against the underlying infection, potentially creating a vicious cycle where the metabolic problem worsens the infection, which worsens the metabolic problem.
Okay, and the second major metabolic crisis,
Yeah.
the one that's really a classic ominous sign of shock progression, lactic acidosis.
Right. This signals a fundamental collapse of cellular energy production. Lactic acidosis in sepsis primarily stems from two overlapping issues. First, there's the straightforward tissue hypoxia. Due to the circulatory failure, the leaky vessels, the microclot cells just aren't getting enough oxygen delivered. Without oxygen, they can't use the highly efficient aerobic respiration pathway in the mitochondria. They're forced to switch to anaerobic glycolysis. This generates only a tiny bit of energy, ATP, but produces large amounts of lactic acid as a byproduct.
But the sources suggest it's not just a lack of oxygen delivery. Correct, there's more to it.
That's a critical point. Even if oxygen is delivered to the cell, the inflammatory mediators themselves, particularly things like nitric oxide, NO, and reactive oxygen species, ROS, can directly damage the mitochondria. They poison the mitochondrial respiratory chain, impairing its ability to use oxygen even when it's present. It's a direct hit on the cell's power plants. So you get this combination of poor oxygen delivery and impaired oxygen utilization, both forcing cells into anaerobic metabolism and churning out lactate.
And that massive buildup of lactic acid lowers the body's pH, which then has knock-on effects, particularly for the circulation.
Yes, the resulting metabolic acidosis is a shock accelerator. That low pH directly blunts the response of vascular smooth muscle to vasoconstrictors, like the body's own adrenaline or the vasopressor drugs we give. Blood vessels become less responsive, promoting further vasodilation, more blood pooling in the periphery, worsening hypotension, and pushing the patient deeper into that progressive stage of shock, ultimately contributing significantly to organ failure.
We touched on the endocrine system with stress hormones, but let's specifically address adrenal insufficiency. It plays a distinct and often critical role in severe septic shock.
Right. The adrenal glands are absolutely vital for the stress response, producing cortisol. In the early phases of sepsis, you typically see a surge in cortisol production as the body tries to cope. But in severe, prolonged septic shock, this response can falter. Patients can develop what's called relative or functional adrenal insufficiency. They just can't produce enough cortisol to meet the massive demands of the critical illness. And adequate cortisol is crucial for maintaining vascular tone and modulating inflammation.
And in the absolute worst-case scenario, that DIC process we discussed can physically destroy the adrenal glands.
Exactly. If DIC is particularly severe, the microthrombin can completely block blood flow to the adrenal glands themselves, causing them to infarct and hemorrhage. This is the classic, though thankfully rare, Waterhouse-Friderichsen syndrome. Losing adrenal function like that, losing that vital cortisol output, dramatically worsens the shock state, leading to refractory hypotension that's incredibly difficult to treat.
So when you put it all together, the systemic low blood pressure, the leaky vessels causing edema, the microclots from DIC blocking flow, the lactic acidosis, the mitochondrial damage, it all converges on the final common pathway: multi-organ failure.
It's the tragic, inevitable endpoint if the process isn't halted. That combination sustained hypotension, tissue edema creating diffusion barriers, microvascular thrombosis cutting off supply routes, and direct cellular mitochondrial injury starves critical organs of oxygen and impairs their ability to function even if oxygen arrives. The heart muscle itself often takes a hit. High levels of cytokines and NO can directly depress myocardial contractility, reducing cardiac output just when you need it most.
And the lungs are almost always involved, resulting in ARDS, right?
Yes. Acute respiratory distress syndrome is very common. That same endothelial injury and leakiness happens in the pulmonary capillaries. Protein-rich fluid floods the alveoli, impairing gas exchange. Ultimately, this systemic onslaught leads to the sequential or simultaneous failure of multiple vital organs. The kidneys shut down, needing dialysis, the liver fails, the lungs require mechanical ventilation, the heart weakens, and eventually, this culminates in death.
That is an incredibly detailed picture of just how devastating this cascade is. Which brings us naturally to Section 4. Updates and advancements, but maybe more accurately, the complexity of intervention. Given how much we seem to understand about the sequence, why has it been so incredibly hard to develop effective targeted therapies?
That struggle really stems from the sheer mind-boggling complexity we've just outlined, but particularly from something we call the immunosuppression paradox.
The paradox being that the initial massive inflammation also triggers a shutdown response. Can you unpack that?
Yeah, it's one of the most fascinating and frustrating aspects, that initial hyperinflammatory cytokine storm we talked about. It doesn't just happen in isolation. Almost immediately, the body launches powerful counter-regulatory immunosuppressive mechanisms trying to put the brakes on. So the septic patient isn't just stuck in hyperinflammation. They often oscillate wildly between periods of extreme inflammation and periods of profound immune paralysis. And trying to treat that moving target is incredibly difficult because a drug that helps in one phase might be harmful in the other.
What's the actual evidence for this shift towards immunosuppression happening alongside the storm?
Well, we see several things. There's often a shift in the T helper cell balance away from the pro-inflammatory Th1 response towards an anti-inflammatory Th2 response. This shift is driven by the production of powerful anti-inflammatory mediators. The big one is interleukin-10, IL-10. It acts like a master brake on the immune system, actively suppressing the production of those early cytokines like TNF and IL-1.
So it's like the body is desperately trying to shield itself from the cytokine storm's damage, but the cost is weakening its ability to actually fight the infection.
That's exactly the trade-off. We also see the body producing things like soluble TNF receptors, basically decoy receptors that float around and soak up TNF before it can hit its real target on cells. And maybe most significantly, we see widespread lymphocyte apoptosis programmed cell death affecting both T cells and B cells. The body is literally killing off its own immune army to try and quell the self-destructive inflammation, but this leaves the patient incredibly vulnerable to the original infection lingering or to picking up new secondary infections.
Okay, so this dual-state complexity really explains the rather grim history of attempts at targeted therapies.
It really does. If sepsis were just a simple linear pathway, too much TNF, block TNF, patient gets better, it would be straightforward. But because you have this simultaneous inflammation and compensatory immunosuppression, most of the clinical trials in the 90s and early 2000s that tried to block single mediators, like using anti-TNF antibodies or IL-1 receptor antagonists, they were largely failures. In fact, some studies suggested that blocking those inflammatory signals too aggressively might have even worsened outcomes, possibly by tipping the balance too far into that dangerous immunosuppressed state.
So the big lesson learned was that just crudely switching off parts of the immune system isn't the answer. It's about restoring balance somehow.
Exactly. Restoring homeostasis is the goal, but it's incredibly hard to achieve pharmacologically right now. That's why the current standard of care, reflected in things like the Surviving Sepsis Campaign guidelines, still focuses heavily on the fundamentals. Get broad-spectrum antibiotics in early and aggressively to kill the bugs, and provide aggressive hemodynamic support, lots of IV fluids, vasopressor drugs like norepinephrine to maintain blood pressure, supplemental oxygen basically, support the circulation and oxygen delivery to buy the body time to hopefully survive the internal chaos.
Now, just to help listeners visualize this clinical decline, can we quickly recap the three general stages that shock, including septic shock, typically progresses through? It gives a kind of framework.
Sure, we usually think about it in three phases. First is the non-progressive or compensatory stage. At this point, the initial insult, the infection, is underway, but the body's reflexes are still working overtime to maintain blood pressure and perfusion to vital organs. Baroreceptors sense the pressure drop, trigger adrenaline release, activate the renin-angiotensin system, all trying to compensate.
And the goal there is just keeping the lights on in the brain and heart.
Essentially, yes. Shunting blood away from less critical areas, skin, gut, muscles, to preserve flow to the heart and brain. Clinically, this often looks like a fast heart rate, maybe some cool extremities as vessels clamp down, although again, remember that septic shock caveat. Early on, the vasodilation might mask this, giving you that warm shock picture initially.
Okay, but if the infection isn't controlled or the body can't keep up, we enter the really dangerous middle phase.
That's the progressive stage. This is where those compensatory mechanisms start to fail. Widespread tissue hypoxia really sets in, forcing that switch to anaerobic metabolism and causing the buildup of lactic acid we discussed. The acidosis then worsens everything, blunting the vasomotor response, causing arterioles to dilate inappropriately, blood pools in the periphery, organ perfusion plummets. This is where DIC often kicks in, and the patient becomes critically ill, totally dependent on ICU support.
And the final stage, if things don't turn around.
That's the irreversible stage. At this point, the cellular injury is just too widespread and severe. It becomes self-perpetuating. Lysosomal enzymes leak out from dying cells, causing further damage. Myocardial function often deteriorates significantly. The gut barrier can break down from ischemia, allowing bacteria to leak into the bloodstream, causing a secondary bacteremic shock on top of everything else. Once a patient reaches this stage, even if we manage to correct the blood pressure temporarily with maximum support, the underlying cellular damage is too great. Organ systems fail sequentially, and death becomes, well, inevitable.
That provides a really clear, albeit grim, trajectory. Okay, let's try to bring this incredibly detailed deep dive towards a conclusion. What are the absolute core takeaways for our listeners about the pathogenesis of septic shock?
Okay, 4 key things to hammer home. 1, remember the core definition. It's a dysregulated host response. The body's reaction is the central problem. 2, think about the 4 pillars of destruction. Uncontrolled inflammation, that cytokine storm kicked off by PAMPs and DAMPs. 3, massive vascular chaos, leaky vessels causing edema, and profound vasodilation causing hypotension. 4, runaway coagulation leading to DIC, that paradox of clotting and bleeding. And finally, 5, crippling metabolic failure driven by mitochondrial dysfunction and lactic acidosis. Those are the core destructive processes.
Right, but despite understanding those pillars in such detail, as you said, mortality is still tragically high, and there are still big unanswered questions.
Yeah.
What's the final provocative thought, the unresolved mystery that keeps researchers up at night?
Well, perhaps the biggest lingering mystery is the precise mechanism of death at the cellular level. We know organs fail: kidneys, liver, heart, lungs. But often, when pathologists examine tissues from patients who died of septic shock, they find surprisingly minimal evidence of actual cell death, of widespread necrosis. There are often no clear morphologic clues, as the sources put it.
Wait, so the organs fail functionally without actually being physically destroyed on a massive scale?
In many cases, yes, the cells just stop working properly long before they physically disintegrate. The real mystery lies in understanding why they enter this state of profound functional collapse. The evidence increasingly points towards severe but potentially reversible dysfunction right inside the cell, particularly alleged in the mitochondria from oxidative stress, NO, and profound energy depletion. The future challenge isn't just about killing bugs or raising blood pressure, it's figuring out how to protect and restore cellular function, especially mitochondrial function, during that septic storm, without dangerously crippling the necessary immune response.
A profound functional collapse rather than outright physical destruction. The real battleground seems to be deep inside the cell's energy factories. That's a fascinating and crucial point. Thank you so much for guiding us through this incredibly complex and critical topic today. And for our listeners, if you want to explore further, digging into the current research on mitochondrial dysfunction and potential protective strategies in sepsis, that seems like the absolute cutting edge right now.
Absolutely. It's arguably the most critical frontier in trying to improve outcomes for these incredibly sick patients.
This has been The Deep Dive.